students, welcome to Biomix. Do you know how are the proteins being transported from the cytoplasm inside the nucleus? What is the mechanism behind it? How the import and export of the proteins are taking from the nucleus? So in this today's video, we are going to discuss about the protein transfer into nucleus. So what happens in the nucleus? The main function is the DNA replication. The DNA replication is taking place inside the nucleus, and for that it requires certain proteins. So the proteins which are required, they are being synthesized in the cytoplasm, and this needs to be transferred inside the nucleus for the whole machinery to be done. The proteins which are present in the cytoplasm, there are different type of proteins. Like these proteins can be transferred to some other organelles also. So how these proteins are recognized? How will these proteins decide that it has to go inside the nucleus or any other cell organelles? So for that, these proteins which are present in the C terminal end or in the N terminal end, they are having certain signal sequences. So what is the signal sequences? They are continuous stretch of some amino acids. Okay, so here for the import as well as for the export, there are different type of signal sequences. Okay, like for example, we will see here for the import of nucleus, that means transfer of protein inside the nucleus, it should have continuous lysine rich sequence. Okay, and for the export from nucleus, it should have leucine rich sequence. Okay, it should have leucine rich sequence. In this way, the proteins are being decided to where to transfer, whether it will go inside the nucleus or it will come from the nucleus. So in general, what happens, the protein synthesis which is formed, it is due to the ribosome as well as the mRNA. So this ribosome, they can go as well as in the bounded form. If it is in the free form and the proteins are being synthesized, these proteins are being transferred into nucleus, mitochondria, chloroplast and peroxisome. So today we will discuss how the transfer of the protein is going inside the nucleus. So before that, we have to understand the structure of the nucleus. So this is the structure of the nucleus. The nucleus which are present, they are having some pores within it. So the nucleus which you are seeing, which we are seeing, it is round in shape, but in this round also there are certain pores which are formed. These are the pores, and from this pores the transfer is this. From this pores the transfer of the proteins are taking place. Transfer of the proteins as well as mRNAs are taking place. So here there are certain proteins in this pore. There are certain proteins which we will discuss. So here this is the complex, and this is the pore which from which the proteins are being transferred. So this is the outer nuclear membrane. This is the outer nuclear membrane, and inside is the inner nuclear membrane. And there are 8 radial spokes which are present in the cytoplasmic side as well as in the nuclear side. There are connections of these spokes. In the nuclear side, this ring which is present, it is known as nuclear pore complex, and they are attached with these spokes. And inside this, they are having here another one connections which are present. Okay, and this portion which is present, it is known as nuclear ring. This is known as a nuclear ring, and inside this portion is known as the nuclear basket. And in the central for connecting these two, there are the central channel. These are the central channels which are present. Okay, and inside this there are certain amino acids which are known as FG, which is known as phenylalanine, and glycine. And this phenylalanine and glycines are responsible for the transfer of the proteins which need to be transferred inside the nucleus, or it will go outside the nucleus. Okay, so here for connecting, there are certain proteins like LBR, hemarine, SUN, which are present in the inner nuclear membrane. And there is a protein which is known as Cas, which is present in the outer nuclear membrane. The SUN protein and the Cas protein which is present, they help in connecting the inner phase with the cytoskeletal filament. Okay, as well as they help in the connecting the lamine. These are the lamines, like you can see the small stretch. These are the lamines. These two proteins help in connecting the lamines as well as the cytoskeletal filament. Okay, this inner nuclear membrane and this protein, amarine protein, this LBR and the amarine protein which is present, it also connect with the lamine as well as the chromatin. These are the chromatins which are present and these are the lamines. Okay, so for the import as well export, there are different type of proteins also which are involved. You can see this and you have to learn this. Now let us see how the transfer is taking place. So for the transfer, it requires certain proteins. Like we can see here, there is a requirement of RAN. Okay, and the RAN which is present, it can be in the form of GDP as well as GTP. Okay, in the cytosolic, in the cytoplasm, in the cytosolic side, it will be present in the form of RAN GDP, and in the nucleus it will be in the form of RAN GTP. Okay, the other thing which we need to understand is the function of the RAN gap. The RAN gap which is present, it will be present in the cytosol. Okay, so what is the function of the RAN gap? When the RAN GTP will come from the nucleus to the cytosol, this need to be dissociated. So this work of dissociation is done by RAN gap in the cytosol. Okay, there is other things which is present in the nucleus, that is a RAN GEF. Okay, which is known as RAN guanine exchange factor. Guanine exchange factor. Okay, so what is the work of the RAN GEF? RAN GDP which is present in the cytosol, it will come inside the nucleus. This RAN GDP will be converted into RAN GTP due to this activity of RAN GEF. Okay, now we have understood that where the RAN GDP is present and where the RAN GTP is present and who is working for the formation of RAN GDP and RAN GTP. Okay, the next thing which we will understand is what is the requirement of this RAN GDP and RAN GTP. So this is the case of the importing of protein inside the nuclear pore complex. So here we can see a cargo protein which is present. This cargo protein means the protein which needs to be transferred inside the nucleus. So it is having a NLS with it. What is NLS? NLS means nuclear localization signal. Okay, it is a signal which is telling that it needs to go inside the nucleus. So for that, there is another protein which will come and bind by recognizing this NLS, that is the importin. Importin is another protein which is responsible for transferring this cargo protein. Okay, so now this protein is being transferred through this nuclear pore complex, and here in this nuclear pore it is having a phenylalanine and glycine rich protein. So phenylalanine and glycine amino acid which will recognize this and it will allow for the transfer of this protein. Okay, so now this protein which is present, it is transferred inside the nucleus, but this protein is bounded with this importin. Now it is binding with this importin. So if it is binding with the importin, it cannot perform its work. So this needs to be dissociated from the importin. So who is dissociating this importin and the cargo protein? This work is done by the RAN GTP. This is a RAN GTP which is present inside the nucleus. The RAN which is present, it is in the GTP form. It is active when it is in the GTP form and it is present inside the nucleus. So this RAN GTP will come and bind with this importin protein, due to which there will be conformational changes, and due to this conformational changes it cannot bind with the cargo protein and there will be dissociation of this protein and the importin. Okay, now this importin which is present, now it is attached with this RAN GTP. This is the importin, this is the importin, and this importin is now attached with the RAN GTP. Okay, this RAN GTP and importin will be transferred again. It will be transferred outside the nucleus. Why it will be transferred outside the nucleus? Because the importin, if it is not recycled, how again it will bring another one protein? Due to this, it is being transferred from the nucleus. Okay, now here it is transferred inside the nucleus. Now when it is transferred from the nucleus, this is in the form of GTP. Importin is bounded with this GTP. Now this will be this. Here the RAN GAP is working in the cytoplasm, which we have discussed earlier. What this RAN GAP will do, it will help in dissociating one phosphate. Okay, so here the RAN which was in the GTP form, it will be converted into GDP form and it will release the importin protein. So now this importin protein can take another one cargo protein with it and it can transfer inside the nucleus. Okay, so here the RAN GDP which is present, it will be again recycled. How the recycling of this RAN GDP is taking place? It is done by another protein which is known as NTF2, which is known as nuclear transfer factor 2. Nuclear transfer factor 2. Okay, what is this? It binds with RAN GDP and it is transferred inside the nucleus. When it is coming inside the nucleus, it will be dissociated, like here, and NTF2 will be dissociated and the RAN GDP will be present. So here inside the nucleus, the RAN GDP does not have any work. So the work of this RAN GDP will be activated when it will in the GTP form. So here the RAN GDP is now converted into RAN GTP. So who is helping for that? RAN GEF is helping. Okay, here the RAN GEF is helping for converting the RAN GDP into RAN GTP form. RAN GEF is known as guanine exchange factor. Okay, so here now the RAN GTP has been formed, which will again bind with this importin and it will allow in the dissociation of the protein and the importin. Okay, this is how the importin of the protein is taking place. Let us now recall again. So what happens inside the cytoplasm, that there is one cargo protein, and for bringing this cargo protein inside the nucleus, there is requirement of a protein which is known as importin. It is transferring through the nuclear pore complex, and inside the nucleus there is RAN GTP, which bonds with the importin protein, and due to which there is dissociation of the cargo protein and the importin protein. Now the RAN GTP which is present, it will be exported from the cytosol. Exported from the nucleus, and inside the cytosol there is one factor which is known as RAN GAP. What it will do, it will help in converting the RAN GTP into RAN GDP and it will separate the importin protein. Okay, now this RAN GDP which is present, it will be transferred inside the nucleus with the help of nuclear transfer factor 2. Okay, now this RAN GDP which has been transferred inside the nucleus, it will be converted into RAN GTP with the help of one protein which is known as RAN GEF. Okay, now let us discuss about the export of nuclear protein. So as for the import, there was requirement of a protein which is known as importin. In the same way, for the export, it requires a protein which is known as exportin. Okay, so here when this will be transferred, here we can see that from here there is transferred, here there is a cargo protein which is present and it needs to be transferred from the nucleus into the cytoplasm. So here the RAN GTP is present. Okay, the RAN GTP, it binds with this cargo protein, the exportin protein. What is this exportin protein recognizes the cargo protein and it binds with it along with RAN GTP works. Okay, and they make a complex. This complex will be transferred through this nuclear pore complex. Okay, when it will be transferred, here the RAN GAP is present in the cytoplasm, and this RAN GAP which is present, it will help in dissociating this RAN GTP into RAN GDP and phosphate, due to which there will be no connection of this cargo protein and exportin. So here the cargo protein will be released inside the cytoplasm and the exportin will also be released. Now the exportin which is present, it needs to be transferred inside the nucleus, and the RAN GDP which is present also, it will be transferred inside the nucleus. So for that it requires another one protein which is known as NTF2. Okay, along with this NTF2, RAN GDP and exportin protein is being transferred, and here there is release of the NTF2 protein and there is dissociation of the exportin protein and the RAN GDP. This RAN GDP which is present, as it was working in the form of GTP inside the nucleus, RAN GEF, what it will do, it will help in the formation of RAN GTP from RAN GDP. This is how the export of this protein is export of the protein is taking place from the nucleus. Okay, now this was all about the transfer of the protein with the help of RAN exporting as well as importing. Okay, now let us now discuss what if there is no RAN present. The next transfer system is a RAN independent nuclear transfer. So what happens inside the nucleoplasm? The mRNA is present. So the mRNA which is present, it needs to be transported outside for the protein formation. So in this case, there is one factor which is known as NTF1 and NXF1 and NXT1. These two factors bind with this mRNA, which help in the transfer of this mRNA inside the cytoplasm, where the protein synthesis will take place. Okay, when these are being transported without the help of any RAN protein, it is binding with this NXF1 and NXT1. And after the transfer, these two proteins need to be again dissociated and the recycles to take place. For that, in the cytoplasm, there is one protein which is known as DBP5. Okay, so this DBP5, with the help of ATP by using energy, it help in the release of the NXF1 and NXT1. And here the mRNA being released and the ribosomes comes and bind in this mRNA, where the protein synthesis will take place, and these two proteins will be transported inside the nucleus. Okay, this was all about the transfer of the protein from the nucleus and into the nucleus. I hope you have understood this video. Thank you everyone for watching this video till the end, and if you like this video, please do not forget to subscribe. Thank you.